So I'm going to show you how to name amino acids and how to put them in a category and organize them as a means of learning them better. So this is the basic structure of an amino acid. It has a carboxylic acid and has an amine group on the end. In addition to that, there's also a hydrogen atom and a R group. This R group basically determines what kind of amino acid it is. Depending what group is here determines any of these 20 amino acids. There are more than 20 amino acids, but for the sake of the MCAT, they want you to know only 20. So what happens is this acid group right here wants to donate its proton, this hydrogen. Now this amine group wants to accept hydrogens. So what happens is this proton leaves, and the formal charge on this oxygen is going to be a negative. And this amine group is going to accept that proton, which turns this into, it gains a proton, which is going to give us a positive charge. Now, this changing makes this called a Zwitter ion. And Zwitter ion is from German origin, and it means hermaphrodite, meaning it can be both acidic and basic. Now that you know the basis of what basically composes an amino acid, let's go to the naming. As you can see, there's 4 categories up here. The first one of them is nonpolar. So for nonpolar, a mnemonic I used to memorize from glycine to proline is GABLIMP. The reason I used GABLIMP was because it was very attractive and it was very easy mnemonic to memorize. GABLIMP doesn't stand for anything, but you can make it stand for whatever helps you remember it better. For me, just this by itself was sufficient. In addition to that, you can see there's tryptophan, there's phenylalanine. So for phenylalanine and tryptophan, I memorized TP. Now TP, in my what I used was toilet paper. And although it is nonpolar, these happen to be aromatic, phenylalanine and tryptophan. However, there's more than just 2 aromatic amino acids. There's another one, and that's tyrosine. We'll get to that later. For polar amino acids, the mnemonic I used was saggy titties. Yes, it's vulgar, but it worked for me. Now I use this phonetically for sag, titties. So that being said, the CAG stands for cysteine, asparagine, and glutamine. Now the T and the T stands for threonine, and tyrosine, and S stands for serine. This is very helpful for me, and it worked. Now for the positively charged amino acids, lysine, arginine, and histine, the mnemonic I used for this was Larice. Now Larice was very helpful because it sounded like Maurice. So if you've seen Beauty and the Beast, you know that Belle's father his name was Maurice. Now there's also Maurice's the store. So if whatever helps you memorize the positively charged amino acids, feel free to use that. But for me, it was Larice. Now, the L stands for lysine, the AR stands for arginine, and the HIS stands for histidine. For the negatively charged amino acids, there's only 2 of them, there's aspartate and glutamate. I didn't necessarily have a good mnemonic to memorize these, I simply just memorized them, but if it helps you, AG, each letter standing for aspartate and glutamate. And if you happen to forget what the abbreviation for silver is, you can always look at the periodic table. You will have one of those on the MCAT. Now, earlier I said there are three aromatic amino acids. There are 3. Tyrosine is the only polar amino acid. Now, there's also tryptophan and phenylalanine. So altogether, those are called aromatic amino acids. Everything else that does not happen to be cyclic is called aliphatic amino acids because they lack a ring structure. Now, something else that might be helpful is recognizing some of the amino acids and where they're commonly used. Methionine, if you've taken genetics, you know that methionine is the most common start codon. It's AUG. Tryptophan, if you're having Thanksgiving dinner, methionine is a compound that's found in turkey, and it oftentimes makes you sleepy. Phenylalanine is oftentimes on the back of the package of, let's say, gum, Coke, any soda for the most part. And sometimes it can actually be fatal if you happen to not have the enzyme to process phenylalanine. Now, in addition to that, something that's also very important to realize is the difference between nonpolar and polar and positively charged and negatively charged molecules and how they interact with the cell membrane. As for nonpolar and polar molecules, nonpolar molecules do not form hydrogen bonds with the membrane, so they pass very easily through the membrane. You could say they're hydrophobic. Now, for polar molecules, they are hydrophilic, which means they want to bind to the membrane, meaning that they have a very hard time passing through the membrane. Now, for charged molecules such as lysine, arginine, and histidine, they have a very hard time passing through the membrane because they want to interact with that membrane and form hydrogen bonds with it. Now, uncharged molecules such as aspartate and glutamate, they can pass very easily. With that being said, oftentimes you'll find the nonpolar amino acids inside the membrane and the polar amino acids on the outside of the membrane because they have difficulty passing through. Thank you for watching my video. If you have any other good mnemonics that you want to share, please put them in the comments. And most of all, please like and subscribe.